So I'd like to give a short introduction about Brain Initiative Cell Atlas Network, BICAN. BICAN is founded by the NIH Brain Initiative with a total of about $100 million per year over 5 years from 2022 through 2027, succeeding the NIH Brain Initiative Cell Census Network, BICCN, from 2017 through 2022. So the BICAN is made up of multiple projects and institutions across the world with complementary capabilities. 4 UM1 centers will have the primary responsibility for generating comprehensive and high-resolution brain cell atlas data in human and non-human primates. A group of UO1 collaboratories will complement and enhance the UM1 centers in creating comprehensive and high-resolution brain cell atlas in mouse and cross-developmental stages. 4 U24 coordinating projects coordinate BICAN operation and establish common data analysis pipelines and the knowledge base of brain cell types. The R24 Brain Initiative Data Archives serve as BICAN data repositories and provide public access to all data and analysis generated by the BICAN. BICAN is also collaborating with 6 NIH neurobiobank sites to establish a diverse postmortem human brain collection and 2 sequencing centers for centralized sequencing contract services. A team of NIH staff will participate in the BICAN activities, providing oversight and program guidance. In 2021, the BICCN, this is an international collaborative effort by more than 300 scientists and at more than 45 institutions across 3 continents, published the first comprehensive census in the motor cortex based on the analysis of the cell's molecular, anatomical, and physiological properties. The effort is a team science on a scale of unprecedented in the neurosciences. And the work has set the stage for comprehensive human brain cell atlases. The overarching goal of the Brain Initiative Cell Census and the Cell Atlas Network is to build reference brain cell atlases that will be widely used throughout the research community, providing a molecular and anatomical foundational framework for the study of brain function and disorders. In October last year, a group of BICCN papers were published in Science and Science Advances on human and non-human primate cell atlases. In December last month, a group of BICCN papers were published in Nature on mouse brain cell atlas. This collaborative effort has provided an unprecedentedly detailed cell census and atlas in a whole mammalian brain, with about 7 million single-cell RNA-seq profiles and 6 million single-nucleus RNA-seq profiles, and about 15 million Murphy single-cell resolution spatial transcriptomics profiles, and several millions of single-nucleus DNA methylation, 3D contacts, and accessibility profiles. As a result, more than 5,000 cell types can be defined molecularly based on gene expression in hierarchical manner. The diverse transcriptomically defined brain cell types can now be mapped to individual anatomical regions in a common coordination framework, providing a comprehensive anatomically annotated cell type map in brain. Furthermore, the epigenomic profiling of single brain cells shed new lights on how the expression of individual genes are regulated in different cell types. Building on these exciting results, BICAN will now expand the horizon to whole human brain across lifespan with more than 100 million single-cell genomics profiling and about 1 billion cells of single-cell genomic profiling, spatial genomic profiling, and moving toward linking single-cell genomic profiling typing to brain function and diseases. In addition, Brain Initiative will fund a new center to study single-cell protein expression signatures on cyto and chemoarchitectural variation between individuals on cell morphology, synaptic structures, extracellular matrix, cell microenvironment, etc., using high-resolution light sheet imaging. Given the expense of brain cell census and cell atlas in generating the large-scale data, about 100 petabytes of single-cell genomics and imaging data, the BICAN program and investigators have put efforts on defining the expected products covering the 5 categories over the whole 5-year period, including published data via brain data archives in raw and increasingly curated annotated data sets, and also standards, SOPs, and foundational references, including coordinate frameworks and publications and software tools. So we'll have more discussion in this workshop on how to enhance the potential BICAN products. The workshop purpose, as laid out here, including foster the development of the data standards for the integration and annotation of single-cell genomics data, and to systemize, automate the process of data to information, to knowledge, and develop pipelines where feasible, and shed new insights on brain cell functional studies, and develop strategies with brain disease research community to maximally leverage BICCN and BICAN data, and finally, to develop a community roadmap for the analysis and annotation of single-cell data. So this is a 3-day workshop, so with a highly compact workshop schedule as laid out here. And I hope this workshop will be successful. And in particular, I want to thank the workshop planning group working hard to make this workshop feasible. Thank you.
Thanks, Yang. That is great to start us off. We just want to have a few remarks from John Nye, the director of the Brain Initiative. And I'll just say John joined NIH in 2020 to run the Brain Initiative, including this set of programs around cell census and cell atlasing. He was an original grantee in the cell census pilot work that started in 2014, and he liked it so much he couldn't stay away. He actually joined us here at the dark side at NIH. So we're of course thrilled to have John, and I'm going to pass it over to you, John, for some remarks.
Thanks, Andrea. I also want to join Andrew in welcoming everybody here. We have, wow, 365 and counting folks on the call right now, which is just terrific. Great thanks to Young and Laura and the entire planning committee for putting together what I'm sure is going to be a really exciting workshop over the next 3 days. Just to give you a little background, the vision for the NIH Brain Initiative as a whole was initially laid out in 2014 when this whole enterprise started. This was laid out in the first strategic plan, the Brain 2025 report, which was authored by a group commissioned by then NIH Director Francis Collins as a working group of the Advisory Council to the Director. That group was co-chaired by Corey Bargman and Bill Newsom. And that plan was really used to lay out our entire vision for what we're doing even today, 10 years, literally 10 years later. And that plan was also updated in 2019 by what we call the Brain 2.0 working groups and their reports. And the neuroscience portion of that was co-chaired by Catherine Dulac and John Mansell. Now, from the Brain 2.0 working group report, they acknowledged the pace of discovery, both within neuroscience and adjacent fields. And they said, geez, this is really a great time to think about investing significant resources to take advantage of these opportunities and to put these resources into large projects that really do or did and do have the potential to transform the field of neuroscience. So the word transform we use a lot, innovation, revolution, but I think these aren't just words of conceit. I think what you'll see from today, this week's workshops, that really great progress has been made, really disruptive progress has been made. In any case, inspired by the Brain 2.0 report, which again came out in 2019, just before I joined officially, in 2021, we launched what we called the Brain 2.0 transformative projects. Again, big aspirational goals, but I think as we'll see, we really are on pace to achieving them. So the first, as we're here today, that I'll just mention is the Brain Initiative Cell Atlas Network. This is the centerpiece of this week's workshop and focuses on developing comprehensive human and non-human brain cell atlases using many different approaches in an integrated fashion. And again, this is the main topic of this week's workshop. Now, as Young summarized for us just now, BICAN builds on the remarkable success of the Brain Initiative Cell Census Network, or BICCN, which recently just published the draft cell atlases of non-human primate and human brains, as well as a comprehensive cell atlas of the adult mouse brain. And we're going to hear a lot about that in a little bit later, or right after I'm finished with my remarks. The second big project is the Brain Connectivity Across Scales, or Brain Connects project. This was recently launched. And the goal here is to develop the tools and technologies required to generate connectivity maps on whole mouse brains at synapse resolution and on big brains at projection level resolution. And that's just getting started. And then the third project is what we call the armamentarium, which is a big fancy word for a big toolkit for precision brain cell access. And here the goal is to develop, validate, and disseminate tools for accessing brain cell types across multiple species. Now, the first two projects will provide the parts list and the wiring diagrams, or the ground truth information for neural circuits. And the resources from the armamentarium project will allow researchers to test hypotheses about the roles of specific cells and circuits in generating behavior in models of both health and disease. So ultimately, we expect these three projects together to inform new precision circuit therapies, including precision gene therapies for human brain disorders. So big aspirations, big goals, big expectations, but I think what we'll see based on the experience in the Cell Census project, this is actually all doable if we put our minds together, and the key word is together, to do it. So for these projects to truly transform the field, it's really critical that the information and resources generated are coordinated and disseminated for access by others in the field. Now, as Young described, the Brain Cell Census project represents a triumph of team science at a scale that's really unprecedented in the neurosciences. It enabled the discovery of amazing proportions, the development of truly revolutionary tools to get those discoveries, and frankly would not have happened, certainly not on this time scale, if it were not for teams figuring out how to work together. So in many ways, BICCN and now BICAN serve as a model for organizing and conducting these large resource-generating projects that ultimately will accelerate discovery of hypothesis-driven projects that both the Brain Initiative as well as other ICs at NIH and other foundations across the world are now really actively supporting. So over the next 3 days, we'll be hearing about some great science that will lay the foundation for the critically important discussions about how best to shepherd and steward these ensuing resources so that they will indeed transform the field and impact disease-focused research and eventually revolutionize the search for new cures. So I would urge us to all keep top of mind that our work is not done until these riches are broadly disseminated and accessible to all. So again, welcome to all. Again, thanks to Young and Laura and the program committee for putting together this week's workshop, and I will hand it over to the next person to continue and dive into the science.
I do want to introduce Honghui Zhang as the Executive Vice President and Director of the Allen Institute for Brain Science in Seattle, Washington. She was one of the first awardees also in the Brain Pilot Program and has been a central leader on this effort since 2014. So Honghui, I'm going to set a timer for 15 minutes and just give you a heads up. That will mean there's 5 minutes left. We do want to have a couple of minutes for questions if we can, and I'll be moderating the Q&A and selecting questions for you at the end of your talk. Honghui?
Okay, thank you, Andrea. I'll share my screen now. Okay, can you see and hear me well?
Yes, sounds good, looks good.
Thank you very much for giving me the opportunity to present today, and thanks, Andrea, for the introduction. I've updated my talk title to Cell Type Diversity and Organization Across the Whole Mouse Brain. The ultimate goal of Brain Initiative Cell Census effort from BICCN to BICAN now is to bridge cell types and brain function. An effective way of doing this is by building a brain-wide cell type map across 4 dimensions, as shown here, across species, modalities, space, covering the whole brain, the entire circuit network of the brain, and across time, from development to aging. Our starting point at BICCN was on the mouse, and using a set of molecular approaches from single-cell transcriptomics, epigenomics to spatial transcriptomics to create a molecularly defined initial framework of cell types across the brain. And as Yong mentioned earlier, we have now been able to do that for the entire mouse brain. From here, then we can extend our investigations into the other dimensions across species, across modalities, and across time. I would say that there are still great challenges ahead of us, in particular, for example, in understanding the cell types, diversity, and definitions across the different modalities. With this kind of effort, we hope that we'll be able to gain a deep understanding of cell types, what they are, what their cellular properties are, how they change under different conditions, and what the conservation and divergence look like across the different species. So again, also as Yong mentioned earlier, the first major milestone we achieved in BICCN was the creation of a multi-model cell type atlas of one single brain region, the primary motor cortex, across several different mammalian species, from human to non-human primate to mouse. And this work is published in Nature in 2021, has allowed us to obtain several organizing principles of cell types: hierarchical organization of the cell types, multi-model correspondence or integration of cellular properties, discrete and continuous variations, and cross-species conservation and divergence. And now, just 2 years from that initial work, we also now achieved another milestone goals, which is to obtain a higher resolution cell type atlas across the entire mouse brain, as shown in this collection of publications in Nature just about a month ago. In this collective work, we achieved comprehensive whole brain coverage using single-cell transcriptomics, single-cell epigenomics, and spatial transcriptomics approaches from a group of researchers, labs within BICCN. In my talk today, I will mainly talk about the transcriptomically defined cell type atlases, focusing on the work that we have done at the Allen Institute, but I will also mention the work from Xiaowei Zhuang's lab and Evan McCoskell's lab, because our findings are very consistent with each other. And I believe that Joe Ecker and Bing Ren will talk about their work on the epigenomics front on Wednesday. So through the combination of 2 large-scale whole brain data sets, single-cell RNA-seq of 7 million cells profiled, and a spatial transcriptomic data set across the whole brain using the MERFISH platform of 4 million cells profiled, we have obtained a cell type atlas, taxonomy atlas of over 5,300 clusters. And the taxonomy is organized in hierarchical manner, with the 5,000 clusters grouped into supertypes, subclasses, and classes. Remarkably, in this whole brain taxonomy, we find that neuronal diversity dominates in the brain, with more than 5,200 of the clusters, neuronal clusters, and the non-neuronal clusters remain as a minority, even though we profiled a large number of non-neuronal cells. And the whole brain taxonomy can be also visualized in this UMAP form, that is colored by different metadata information. What we can see from this presentation is that the cell types are defined not just by their molecular identity, such as the neurotransmitters they express, but it can also be defined. It's very much driven by their regional specificity. And in fact, the 34 classes that we have defined, especially the neuronal classes, are really a combination of their molecular identities and their regional specificity. Furthermore, by registering our spatial transcriptomics MERFISH datasets to the CCF and perform quantitative analysis of the spatial distribution of all the cell types in the brain, we find a high degree of correspondence between the molecular identities, the transcriptomic identities of the cells, and their spatial specificity, as shown in this graph, and shown only at the class and subclass level. The other two studies from Xiao Wei and Evans' lab also showed that the similar kind of high correspondence between transcriptomic identities and spatial specificity. Furthermore, we also identified an interesting dichotomy in cell type characteristics in different brain regions. Namely, in the largely ventral part of the brain, mainly coming from hypothalamus, midbrain, and hindbrain, we find that there are more numerous, a much larger number of clusters or cell types in those areas. And those clusters tend to be a lot smaller, containing fewer number of cells. And the clusters are more similar to each other based on the number of differentially expressed genes between each pair of clusters. And also the clusters in those regions are spatially more confined into restricted locations. The dorsal part of the brain, on the other hand, especially in cerebral cortex or the pallium structure, the thalamus and the cerebellum, on the contrary, we find that there are fewer number of clusters or cell types. The clusters tend to be larger, containing more cells, and they are highly divergent based on the number of DE genes between each pair of clusters. And spatially, they are also more widely distributed. We hypothesized that this dichotomy between the dorsal and the ventral parts of the brain probably originated in evolution because of the differential functions of those two different parts of the brain, with the ventral part of the brain, hypothalamus, midbrain, and hindbrain, mainly carrying out survival functions like feeding, reproduction, metabolism, homeostasis, regulation, and things like that, and that are essential for the survival of the animals so that they may be subject to more evolutionary constraint. So the cell types within those regions have not changed much, even though they are older, more ancient structures. The dorsal part of the brain, especially cortex and thalamus, for example, they mainly carry out adaptive function to allow the animal to adapt to new environments and to gain evolutionary advantage. And for that reason, maybe that's why the underlying cell types have evolved much faster and diversified much faster. So this is an interesting hypothesis that can be derived from this kind of unbiased analysis of large-scale comprehensive data sets. We also wanted to understand the relationship between different cell types, distance relationships with different cell types. And we did this by looking at examining the gene expression correlation a matrix using different kinds of genes. This is a correlation matrix generated by 500 of the most significant 500 transcription factor marker genes showing the gene expression correlations. And here additional matrices showing similar kind of correlation matrices, but using about 500 functional genes, about 800 adhesion molecules, and all 8,500 marker genes that have been identified in our whole brain transcriptomic taxonomy. So the difference among those matrices actually is quite remarkable. It shows that the transcription factors with just 500 genes are actually able to distinguish, showing the differences between different cell types at different levels much better than the other families of genes, and then even the entire marker genes, 8,000 marker gene set. So transcription factors are best able to distinguish the cell types at different hierarchical levels, not only at cluster level, but also especially at higher levels, classes and subclasses level. So in fact, because of this, we have used mainly transcription factors to define the hierarchical organization of the cell types across the entire group. Furthermore, we are also able to identify transcription factor gene modules, 52 gene modules shown here, that comprise a transcription factor code that defines and specifies the 5,000 clusters and their hierarchical organization at class, subclass, and supertype level, suggesting that transcription factors are really a major gene family defining cell type identity. Okay, now in the next couple of slides, I'll just quickly go over what the different families or neighborhoods of the cell types that we have subdivided in order to further understand the extraordinary diversity of cell types in the brain. Here, we defined 7 major neighborhoods based on our major brain structures and generated sub-embedded UMaps and examined the spatial distribution of the cell types within each of the neighborhoods. And showing that even as we dive deeper into the specific structures of the brain, we continue to see this extraordinary diversity and specificity in all different regions. Pallium glutamatergic, subpallium GABAergic, and hypothalamus extended amygdala areas shown in this slide. Thalamus and midbrain, hindbrain, glutamatergic, and GABAergic neuron types shown in this slide. In addition to the neuronal types with extraordinary diversity, we also observed very interesting heterogeneity and diversity in non-neuronal cell types, even though they are much fewer than the neurons. The diversity is particularly announced in the astrocyte and ependymal cell classes. And here, just using 2 slides to show the beautiful transcriptomic diversity and their corresponding spatial specificity of the astrocytes down to cluster level, and ependymal cells. Again, down the hierarchy into cluster level, showing the specific location, specific expression of individual ependymal cell types that are in different parts of the ventricles of the brain. Okay, without getting into much detail or further move along, we also reveal extraordinary diversity in the neurotransmitter contents in the different neuronal types across the brain. And this slide shows all the cell types containing modulatory neurotransmitters, dopamine, acetylcholine, norepinephrine, and serotonin, and histamine, and showing that not only the diversity among the modulatory neurotransmitters, but also their co-release, co-expression, or co-release patterns with glutamate and GABA. And we can see all kinds of combinations in different cell types or clusters. This is also illustrated in Xiaowei's spatial transcriptomics Murphish dataset with the very specific clusters expressing a specific set of neurotransmitters found in a very specific location of the brain, for all kinds of neurotransmitter types: dopaminergic, serotonergic, histaminergic, noradrenergic, cholinergic, and etc. Furthermore, Xiaowei's data and our data, and Evans' data as well, also showed the extensive, also extraordinary heterogeneity and diversity of the neuropeptide gene expression across the different cell types, across different parts of the brain. Some neuropeptides are highly specific to only a few, 1 or 2 clusters in the brain, whereas some other neuropeptides are widely expressed in many different kinds of clusters across the brain. Xiaowei's group and we at Allen also are able to use computational approaches to impute the entire transcriptomes from the single cell RNA sequencing data into the Murphish space, so that we can now spatially visualize the gene expression patterns of any genes in the transcriptome, even though the genes may have not been profiled directly in the Murphish experiments, the 500 gene panel or the 1,100 gene panels the Xiaowei's group have used. And here are some examples showing beautiful.
Sorry, Honghui, about 4 more minutes. Apologies.
Okay, I'm almost done. Beautiful examples of imputed gene specificity in comparison with the original in situ hybridization expression pattern of the genes themselves, and high degrees of correspondence. So here's my last slide, showing, trying to make a point of the high degree of consistency across the different data modalities that we have generated in this consortium. And this T-SNE plot shows the more than 5,000 clusters identified in the single nucleus RNA-seq-based cell type atlas generated from Evan McCoskell's lab. And using a hierarchical mapping method, we showed that the 5,000 clusters from Evan's atlas corresponds quite well, amazingly well. It is shown in this confusion matrix with the 5,300 clusters derived from the single cell RNA-seq data from the Allen Institute. So with that, I just wanted to conclude by summarizing what I have told you about today. From our earlier single cortical region studies, we have found the several principles of cell type organization, including hierarchical organization, coexistence of discrete and continuous variations, and similarity and difference across species. Now with the transcriptomic cell type atlas for the entire mouse brain, we identify extreme diversity and complexity and distinct features of cell types in different brain regions. Transcriptomic types often have regional and more fine-grained spatial specificity, revealing an integrated molecular and anatomical organization of cell types across the brain. The relatedness between cell transcriptomic types, we think, likely reflects the developmental origins and or the evolutionary homologies. And in this regard, transcription factors really carry that historical developmental and evolution information, and they are the major determinants of cell type classification even in the adult mouse brain. We identify extraordinary diversity and heterogeneity in neurotransmitter and neuropeptide expression patterns, and we expect similar things regarding other signaling molecules and cell adhesion molecules, and thus illustrating a myriad mode of intercellular communications. And finally, I showed very quickly that we observed a high degree of consistency across different data modalities in this particular collection of work between single cell RNA-seq, single nucleus RNA-seq, spatial transcriptomics, and also methylation-seq and attack-seq, giving us high hope that we will be able to generate an integrated cell type atlas across modalities for the whole mouse brain. With that, I just want to thank a large number of scientists, especially for my talk, scientists, technologists, and managers at the Allen Institute in making this work happen. And also thanks our collaborators, Xiaowei Zhang's lab, as well as funding from the NIH Brain Initiative. Thanks for your attention. And I hope I have a few minutes to take questions.
Thanks, Honghui. That's a great overview of the mouse atlasing project, which was amazing. We do have a few questions in the chat, or sorry, in the Q&A, so I encourage others to ask. It looks like Dr. Yong Yao is typing an answer to one of them, which is more of a technical clarification. So I'm going to move on to one of the questions. How many mice were used to generate the single cell data sets? And were there across institution validations for the RNA-seq cluster? So what sort of validation did you do?
Yeah, that's a great technical question. That's important for us. So because we use single cell RNA-seq, we have to use a fresh mouse every day for dissection and isolating live cells for sequencing. So we used more than about 320 mice for this study, 780 10x scRNA-seq libraries used. So it's not coming from a few number of animals. It's really a large number of animals, all 67 black 6 background. We use the stringent criteria for QC. In terms of cross-institutional validation, I wanted to use the Murfish data as a great example as validation. Both our data and Xiaowei Zhang's lab, we generated altogether 7 whole brain level data sets using different gene panels completely independently. And we obtained exactly the same, almost exactly the same spatial location for every cluster, almost every cluster, certainly supertype level, subclass level, high degree consistency. But for a large number of clusters, more than 90% of clusters, we also obtained the same specific spatial location. So we think that's a really good cross-institutional validation. But at the same, and also the last one I showed, the corresponding between our clusters and Evan-McCosk's clusters is another way of validation, but we're continuing to do that. Compare our data sets with other published data sets as well.
Yeah, very important. Thanks. We don't really have time to move on to more questions, but I just want to note that in the Q&A, there's a lot of interest in transcription factors. And this is a really interesting piece that has been revealed through this resource. I want to remind all of our attendees that the data sets are public, they're available. We really encourage anyone who's interested in any aspect of this to get your hands dirty, get into those data sets and start doing your own analyses. That's exactly why this resource was created, to be broadly available and to answer some really interesting questions around biology. So I'm going to leave it at that, but we are collecting these questions, and hopefully those of you online can also help answer these questions live. So Conqui, I'm not sure if you can, I think you should be able to see the Q&A. You might want to get into there.
I can try to answer it.
Answer some questions as well. Okay, so I think we will now move on to our next speakers, and I'm thrilled to introduce two folks who grew up in the atlasing program, as far as we're concerned, welcoming Aparna Baduri and Tomas Nowakowski. So both Tom and Aparna were trainees in some of the original sets, and they have gone on to do fantastic independent work as well. So Tom is an associate professor at the School of Medicine at UCSF, and Aparna joins us from UCLA as an assistant professor. And I won't spend any more time except to turn it over to the two of you. Again, I will give you about 15 minutes. I'll give you a 5-minute note call, and then you can wrap it up and hopefully answer some questions as well. So on to the two of you.
Great. Thank you so much, Andrea. I'm going to share my screen. Can you see my slides? Can you hear me okay?
Yes, all looks good.
Great. So it's a real pleasure to join this webinar and discuss together with Aparna an update and provide an update on the progress on understanding, on mapping the developing brain atlases. We wanted to both provide an inspiration and justification for why we need to better understand the developing brain, what that means potentially for our ability to understand the etiology of developmental disorders, to provide an update of where the Brain Initiative Cell Atlas Network program is currently, and to highlight some of the unmet needs. And so thanks to Hongqui's beautiful introduction, we really have been appreciating and mesmerized by the tremendous diversity of cell types that exist in the adult human and non-human brain. And this inspiration obviously dates back to the days of early anatomists who have been classifying cell types purely based on what they look like and recognize the sheer complexity of morphological types. Thanks to the work that Hongqui described, we now know that the adult mammalian brain can contain as many as 5,000 or more distinct transcriptomic types, and the transcriptomic definitions have really provided an anchoring for our understanding of how diverse cell types in the adult brain may be. But this is, of course, only just a starting point towards understanding where do these cell types come from during development, and what are their developmental trajectories that set up this tremendous complexity. And one of the reasons why we want to understand how these cell types are created during development is because there is a whole range and a whole spectrum of neurodevelopmental, behavioral, and intellectual disorders that affect quite a large number of individuals, young individuals in industrialized countries. And we're only just beginning to scratch the surface on understanding what the diversity of cell types may be in the developing brain. So starting from early anatomists, it has been sort of perceived, a common concept has been that the diversity of cell types in the developing brain may be significantly reduced compared to adult brain. And a lot of those ascertainments have been made based on the descriptions of cellular diversity that was purely based on morphology, really. Thanks to the initial work from the Brain Initiative that has supported some of the early single-cell RNA sequencing transcriptomic studies, have almost immediately revealed that even despite these apparent reduced morphological complexity of cell types in the developing brain, there still exists quite a striking molecular diversity. So using single-cell RNA sequencing, we can discover new cellular types or cellular states that exist in the developing brain that wouldn't be apparent from basically looking just purely at morphology. And the reason why this is important is because it provides us with an insight into the molecular cell types that may be present during development. Why is that important? One of the reasons why this is important and will advance our understanding of the etiology of neurodevelopmental disorders is because thanks to the advent of human genetics studies, we now have identified, supported largely by the NIH, but also by Simon's Foundation and other foundations, we have now defined or identified genes with genome-wide significance excess of damaging mutations in patients who have been diagnosed with neurodevelopmental psychiatric disorders, including ASD and related neurodevelopmental disorders. And one of the goals for understanding the possible etiologies of these disorders is to identify where and when during brain development these mutations or these genes are most important. I.e. to identify the select vulnerabilities of cell types and developmental periods in brain development. And even some of the early studies have highlighted that prenatal periods of brain development may be particularly relevant and vulnerable to these developmental disorders. And these periods of development highlighted here correspond to some of the processes when the neurons and glia are just being generated. And a lot of these insights could be gained from even prior to single-cell analysis, advent of single-cell data sets. But by integrating the genetic enrichment, gene expression enrichment analysis with single-cell atlases, we can actually now begin to pinpoint specific cell types which are enriched for expression of high-confidence neurodevelopmental risk genes, providing an insight into the possible vulnerabilities of the developing brain to damaging mutations that are found in patients. So this is one very concrete application of how cellular resolution atlases can provide us with new insights and new hypotheses to how neurodevelopmental disorders might occur. Another practical application of these data sets that I wanted to highlight has to do with evaluating the fidelity and robustness of in vitro models of human brain development. As some of you may have heard, stem cells derived from skin fibroblasts that may be derived from adult individuals and including healthy individuals as well as patients with neurodevelopmental or psychiatric disorders can be derived at scale using the advances in stem cell technologies. And these stem cells can be subsequently differentiated in vitro into what is known as brain organoids. And these are in vitro models that we can use to study the etiology of neurodevelopmental disorders thanks to the advent of gene engineering technologies, as well as technologies that allow us to perturb specific gene expression that you will hear about from Aparna. And one of the benefits of the data sets that the Brain Initiative has created is that we can now quantitatively compare the fidelity and robustness of gene co-expression networks that emerge in these in vitro models and how they compare to the developing human brain in vivo. And these comparisons can, in a quantitative and rigorous way, evaluate and highlight the remarkable similarities, but also some differences between those in vitro models and developing brain. So these are some of the very concrete and practical utilities of these data sets that we wanted to highlight. And just to summarize where we are particularly with the work supported by the Brain Initiative Cell Census Network. There's been a number of pilot studies highlighted over here, supported by the Brain Initiative that were overwhelmingly focused on the developing mouse brain, but with some limited number of studies that embarked on mapping the developing non-human primate and human brain. And a lot of these studies have now been concluded with an advent of a number of, and have resulted in a number of publications, some of which are listed here, many of which have been published in high impact factor journals, which I recommend that you follow up on and read, which are very exciting. And now that we embark into the next phase of this effort that you will hear about more from Aparna in a second, that have really been pioneered now by the investment from the Brain Initiative Cell Atlas Network, which seeks to create more comprehensive atlases of the developing human, non-human primate, as well as comprehensive data sets from the developing mouse brain. And some of these milestones and goals are listed over on this slide, and we hope to, in a few years, provide you with additional updates or how these data sets can become more useful and advance our understanding of the developing brain. With that, I will hand over to Aparna, who will speak a bit about her lab's efforts to integrate and make sense of this data and put it to practical use. Aparna, over to you.
Thanks, Tom. Thanks for that amazing introduction. And I think that one of the goals of this section of the talk is really to provide an example of what Andrea was talking about, where we can really take data that's out there and try to integrate it to understand key biological processes. So this is one of the things that we've been trying to do, and is also something that we're trying to extend as part of a joint analysis to additional data sets. So this is hopefully just the first step of these types of explorations that we are planning to do through our UM1 funded work. As part of the BICAN initiative. Our goal for this project, which is led by Dr. Patricia Nano, who's an incredibly talented postdoc in my lab, was really to create a meta-atlas of the developing human cortex. And the idea was to create an integrated analysis of 7 transcriptomic profiles where we integrate data sets, many of which were funded by the Brain Initiative and some that were independently conducted by other labs, focusing primarily on developmental periods that are embracing the peak stages of neurogenesis during the 2nd trimester, but also that flank this time period depending on the data set that wasn't included. Based on this conventional integration, you can see that the major cell types that we would expect to exist during human brain development and cortical development specifically are represented. And what Patricia sought to do was to use an orthogonal strategy to identify novel gene co-expression networks that would allow us to identify biological processes that correlate and are parallel to some of these cell types in order to really address some of the issues that Tom raised, where we know that there are transient cell types and that they're important biological processes, but we're still learning to understand how they connect to the cell types that we see in the adult. So briefly, the method that was used was we individually analyzed each of the 96 individuals and performed unsupervised clustering for each individual. This allowed us to really correspond to gene signatures that were variable within an individual, and then to avoid batch effects. This also allowed us to integrate between individuals to find the strongest signal that allowed us to make meta modules with biologically meaningful information rather than technical variation. Patricia went on and annotated these modules, and many corresponded to interesting biological processes, some of which we believe are related to processes such as cell division or cell fate, and then others where it's unknown, but we're hoping that using some of the patterns of activity for these modules, we can better understand their role in development. We use some positive controls. So for example, modules related to vascular function were expressed in vascular cells, similarly for microglia and immune function, as well as some of the glial subtypes, which gave us confidence in the specificity of our modules, as well as our ability to then extract novel information from other modules within the data set. Our goal was then to identify modules that could give us insight into potential biological processes, such as the initiation of cell type specification, the refinement of these identities, as well as a little bit of mechanism, which I'll talk about at the end. Beginning with initiation of cell type specification, we focused on module 156, which based on our annotations was related to radial glia biology, neuronal activity, and response to stimuli. And as you can see by the module activity, was broadly expressed in radial glia, though interestingly, its activity was increasing through radial glia across development. What was particularly exciting to us, and this is one of the ways that we sought to leverage Brain Initiative data, is when we looked at the module activity for this module in adult data that was generated by the Allen Institute, we saw a specific expression within glial populations. We then wanted to see whether there was any evidence that we could really represent this particular module with some spatiotemporal specificity. And indeed, looking at the co-expression of two of the genes, PIDLM5 and QKI, we saw that they were specifically co-expressed at later stages in gestational week 20 ventricular zone samples, suggesting that this module is active in progenitors that are later on in development. Well, we haven't yet performed experiments where we can mechanistically test what happens when these programs and this module is eliminated from the cells. We think that this is consistent with the idea that there are certain biological processes that yield specifications, such as towards a glial fate, that we can find from these types of data sets, and that this type of an analysis can also be additionally hypothesis generating. When thinking about the refinement of cell type identities, we focused on neuronal modules that were somewhat broadly expressed across neuronal populations in our developmental data set. And one thing that's been a challenge in the field is really identifying which of the cells within broadly upper or deep layers would become individual subtypes. And although there was some subtype specificity for these three modules, 134, 94, and 189, they were broadly expressed across a number of excitatory neurons. And their module activity was increasing across newborn neurons and excitatory neurons across development. However, again, when we leveraged the adult data, we saw that for some of the modules, and most notably here, you can see it's striking for 134, there was layer specificity to layer 4. And we, again, used immunostaining to look for co-expression of some of the module genes that are expressed within module 134, and saw that they're co-localizing in upper layers during development. And we can zoom in into these two panels and see that there's more co-localization in the upper layers compared to the deep layers, and that the co-localization sits right above CTIP2, which is typically a marker for deep layer function. We also wanted to pursue whether there were any of these modules that could give us some mechanistic insight into cell type specification. One of the modules that was really exciting to us, which was this module 20, which we saw to be expressed in broadly across our neuronal populations, and that was increasing specifically in deep layer identity cells, but not in upper layer. More specifically, when we looked at this module 20 within the adults data again, which has been immensely helpful in contextualizing our analyses of development, we saw that there was specificity to these deeper layers, and that across layers 5 and 6, there was specificity to the subtypes annotated by the Allen Institute as FESF2 transcription factor marked subtypes, building on some of the, connecting to some of the ideas that Honghui was presenting in her talk. What was interesting though, is that this module doesn't contain FESF2. However, in development, we saw that FESF2 expression was preceding module 20 expression and activation, which suggested to us that possibly module 20 is regulated by FESF2. Indeed, when we leveraged data that existed from FESF2 ChIP-seq, we were able to see that more than half of module 20 was regulated by FESF2, and this included the transcription factor TSHC3, which was the only transcription factor found in module 20. And as you can see, there was some concordance of TSHC3 expression in the subtypes that we were interested in, as well as correspondence in its activation in timing with module 20. So we wanted to test—
just giving you a 3-minute warning or so.
Perfect, we're just about wrapping up. So we really wanted to test whether or not TSHC3 and FESF2 could be involved in the specification of this. And again, as Tom mentioned, we've carefully compared organoid data to the primary data from the developing brain, so we understand some of the strengths and weaknesses of the model. So we sought to use the system by knocking down TSHC3 and FESF2 and perform single-cell RNA sequencing on the knockdown cells compared to the non-knockdown cells as determined by M-cherry positive expression, where the hairpins had M-cherry, so we could really perform a controlled experiment in the same environment. With the single-cell RNA sequencing, we were able to identify cell types that we typically see within organoids, and we're able to contextualize them compared to primary cell types, and broadly the cell types were corresponding to what we would expect. We saw that there was a modest depletion in module 20 activity with the FESF2 and TSHC3 knockdown, but that this corresponded to a substantial depletion of deep layer neuron proportion, suggesting that there's a cascading effect where each of these transcription factors has a role in controlling module 20 activity, but then this impacts deep layer specification. We were really excited about this because this was an example of how we were really able to use data right up until the last experiment to really develop a model and then test it using models that we have for human cortical development. And the model that we think that we have resolved on is FESF2 activation precedes TSHC3 activation, which then gives rise to module 20, which is important for this in-between specification event that happens between the earliest stages that we were looking at during peak neurogenesis and the adult deep layer FESF2 neuronal specification. So this strategy has allowed us to start to understand how we can bridge the gap between the developing and adult human cortex. And we also have not only mechanistic insight, but also a resource if other people want to look into this. This is also a bioarchive if anyone's interested. And we think that this is also a strategy to integrate other data sets, which is one of the things that we are seeking to do as part of our UM1 moving forward. When we think about these integrations, I think that especially across development, there's a number of challenges that continue to persist. And the image that I've shown here, and I'm sorry, I think it's not sharing the recognition, but from Rong Fang, Hua Wang, and Asnad Sistan, you can see that we are seeking to really go from the molecular level to actual physical structures using spatial transcriptomics to make the link, and then to link that all the way back to MRI data and really coordinate framework integration. And so this is one level of integration that we're seeking to do as part of our project, but we're also thinking about how we can do this between species, and then importantly, between developmental stages and modalities. And so I think these are different axes of integration that we're hoping to really get a handle on in the coming years. With that, I'd like to thank the members of my group, the members of our Brain Initiative project, and all of the funding sources, and especially the BICAN funding. And thank you for your engagement. I'm happy to take questions.
Great. Thank you, Aparna, and thank you, Tom. We're being super efficient in the Q&A. So it looks like a lot of these questions are being answered along the way by colleagues and just people who are interested in the biology. So I don't have a question. I just want to say how this is such a great example of how you take different data sets that are really generated, as a resource, and people understand the data and really look for these biological insights that may not have been possibly before at the level of resolution that you all are able to do. So that's great. Oh, we do have a question. So I'm going to go ahead and read that. They say, thank you. This is exciting. What was the temporal window for the FESF2 mediated regulation of module 20? Does the intermediate transcription factor, TSH, take over at some point? Is that obvious to you?
Yes. So I don't know if it's obvious. I think it's a good question. And I think that we're seeking to understand this timing. But what we were seeing is that during earlier stages of neurogenesis, especially in the human, and this is actually something where the temporal expression was a bit different than what we were seeing in mouse data, was that FESF2 was active early in those neurogenesis stages, and then its expression declined. And then TSHC3 and module 20 were coming up. We are not fully sure if TSHC3 takes over entirely. Its expression also decreases after a wave of expression, but the other module 20 genes then consistently increase. And so we think that it might be kind of a handoff where it needs to get the ball rolling, but then the transcription factors aren't as essential. And I see that Tom has his hand up.
Oh, yes, I just had so this was fantastic. Thank you, Aparna. I wanted to ask another question, which is, how do you think about integrating the data sets across species? Obviously, when we think about the human brain and the mouse brain, there's quite a difference in size and hopefully function as well. So what do you think are the developmental differences that have driven the dramatic expansion of the human brain, and how can the data sets that you're creating, how could they lend themselves to our understanding of what made us human?
I think that's a great question, Tom, and I think we could toss it back to you as well. I know you've thought about these questions extensively, but briefly, I'll say that some of the data approaches that we're using, I think, allow us to get some crosstalk between data sets of different developmental stages and different species. I do think that there's a lot of questions to be answered in terms of, are we really thinking about different species and different genomes? Are they consistent, and are we introducing any biases? But those technical questions aside, I think that there's a lot of ways that we can start to use the data to compare timing and try to understand how at a process-specific level there are differences, whereas not all of the timings are going to be moving at the same pace. And then I think that, there's a lot of work that folks in the Brain Initiative are doing to understand really what this means for how evolution proceeded. And so I'd also ask you to chime in on that same question if you have any additional thoughts.
No, I think this is really important and highlights some of the vulnerabilities to potentially has the potential to highlight vulnerabilities of the human brain to various types of neurological disorders. Andrea, do we have time for one more question?
One more question. We are bleeding into our break a bit, but if it's quick, we can do it.
Yes, there is a question in the Q&A. When do meta-analysis, when would you recommend doing meta-analysis? How do you control for batch effect, for example, data combined from different researchers?
Great question. And this was actually a motivation for one of the reasons that we sought to use the strategy that we did, where we're looking at each individual and then taking variation that was observed within that individual and applying it to broader patterns that we were seeing. One of the reasons for this is, many of the batch correction approaches that exist out there right now are excellent, and they do a good job of integrating, especially at the cell type level, but we were concerned about whether any of the gene co-expression patterns would get diluted out. And so that's why we approached the strategy that we did here. I think that the batch effect is always a concern and something to be thought about, but I think that what we're learning is by being creative in the way that we address it and acknowledge it, and then look for things that could be technical artifacts, we can always still make use of data that has been generated by a lot of people. And at some level, there's power in numbers, and having more data sets from more individuals can give us more insight into really understanding the biology.
It is now 12:55. So my name is Amanda Price. I'm a program officer at NIMH, and it is my pleasure to introduce the first panel on brain cell atlas data integration and annotation. This panel is moderated by Drs. Wosheng Yuan and Jesse Gillis. Dr. Yuan is a professor of computational biology at BICCN School of Medicine at Mount Sinai and a faculty member of the BICAN Genomics Institute. Dr. Gillis is associate professor in the Department of Molecular Genetics at the University of Toronto. So I'll now hand the meeting over to Drs. Yuan and Gillis.
Thanks, Amanda. GC and I are splitting moderating duties, so I'm taking the first one, and then GC will follow with the second one. And the way we've organized our topic, which is brain cell atlas data integration and annotation, is into three sort of mini sessions. The first principally focused on where we are, kind of from the past to the present. The middle session more on specific data problems that are currently occurring in development and new modalities. And then the third session focused on new technologies and approaches in the future. What directions are we taking? And so for each session, we have two very short presentations or talks that we hope will provoke some questions and thoughts, not specifically to the talks, but to the topics. And so we're beginning with Daifang Wang. And I won't give long introductions, just from the University of Wisconsin, and I encourage you to look up his very interesting work over the years. Daifang, if you want to share your screen.
All right, thank you, Jesse. Yeah, can you see my screen?
Yep.
All right. Thank you, everybody. So first, I want to thank NIMH and the Brain Institute for organizing such a wonderful workshop about data. I'm happy to give a first short presentation about our first topic about cell types. So we know that Brain Initiative has generated a lot of multimodal data to characterize the single brain cells at different aspects. For instance, like RNA-seq, single-cell RNA-seq and tag-seq measured gene expression, the chromatin accessibility, and the PET-seq can give people the electrical physiological features or morphological features. So using those multimodal data, actually, we can integrate those data and map them onto some latent space and cross the cells on this latent space, and eventually get cell clusters. So those the cells clustered together imply that they share very similar multimodal features and the defining potential like a cellular state. So however, what we don't know actually is that are those cell clusters corresponding to a particular cell type or a specific cellular function? So for instance, so definitely we can map our data onto some reference data, for example, transfer the labels to annotate cell types. Those cell types actually defined by some prior knowledge or particular features such as marker genes. However, those marker genes may not fully represent the underlying function of the cell clusters. So for instance, like we know like a gene regulatory network fundamentally control the gene expression, and those networks can link the transcription factors to the regulatory element like enhancer and promoters to control target genes. So but the regulatory networks can change across different conditions like disease types or developmental stages, such as like switching the TAs or the enhancers for different cell clusters. Even they express very similar marker genes. So that's why we need to leverage additional techniques to better understand cell types or cell functions. So here, I just give some like idea. For example, we can leverage machine learning to integrate and analyze the single cell multimodal data to see if we can better understand cell types or cellular functions. Definitely, the community has published a lot of great machine learning approaches to do this. So here, I just use my some recent work in my lab actually to summarize several ideas that we can leverage machine learning. So for instance, we can definitely use like a manifold learning to align the multimodal features and map cells actually from different modalities onto the same latent space. And found like cell clusters on such a shared same latent space. So so the clusters eventually suggest some like cross-modal cell types with a similar multimodal features instead of single modality like gene expression. Also beyond individual features, so we can also infer some multimodal feature relationship, such as a gene regulatory network, so which may better define the cell types. So for example, to see how the network defines the cell types. And also we know that the multimodal data is not always available. So we can also use the machine learning to impute missing modalities. So basic idea is that we can use the available multimodal data to train some machine learning model to infer one modality from the other, and then use the apply the pre-trained model to the single modality data and impute missing modalities. And eventually, we can input population level multimodal data to some machine learning model, like deep learning model, to prioritize the genes, gene regulatory networks, and the cell types for disease types and the clinical phenotypes. So not just to learn some black box model, but also make the model biological interpretable. So yeah, so due to the time limit, I stop here, and I look forward to the panel discussion. Thank you.
Thanks, Daifeng. That's great. And moving on to Josh Wong at Duke University for talk I'm looking forward to as well. You're muted, Josh.
Oh,
Yep,
okay. Good afternoon. Hope everybody can hear me now and see the slides. Yeah. So as outlined by Yong at the very beginning, one of the goals here is to go from data to knowledge and to ultimately insight and understanding principles. So one of the challenges coming back to understanding and defining cell types, and to achieve a overarching definition that will integrate multiomics data sets that is now in massive amount scale and coming very quickly. But to integrate this with multimodal phenotypes, we have heard anatomy, morphology, physiology, and ultimately the goal is circuit function. But all this based on biological mechanisms, because we believe there is a biological mechanism that goes from gene expression programs to cell phenotypes and circuit function, and principles that involve developmental lineage, trajectory, and even evolutionary history. And all of these, hopefully beyond the sort of operational management style definition by statistical parameters and in often arbitrary scenarios. So just to facilitate the discussion, I'll propose one definition that we have been thinking about, and it's a working hypothesis. So this is what I call a communication element definition of neuron types. And in this regard, the neuron types can be specialized communication devices which can be defined by their unique connectivity patterns, input-output transformation that actually may be shaped by specific transcription signatures of gene batteries that is the output of gene regulatory programs that ultimately is rooted in the epigenetic landscape. So let me unpack this a little bit. Communication element is defined by its connectivity, anatomical connectivity, what's the presynaptic and postsynaptic elements, and also beyond how they transform physiological input to output in the context of circuit computation. So connectivity is an overarching anatomical feature over location and morphology. There have been a lot of emphasis on morphology, including large-scale single-cell morphology reconstruction, striking shapes. But the goal of neurons is not to enter a beauty contest, I think we all agree, but to really connect to the correct presynaptic cells with extending dendrites and reaching its proper target by the very long axon. So this is a feature that is, I think, essential, but difficult to characterize. And the other is that we use intrinsic physiology, synaptic properties in slice to characterize physiological properties. But we know that this is highly, highly limited. Ultimately, the goal of physiology is to perform input-output transformation in the context of circuit computation and behavior. Again, a very difficult feature to measure. However, there have been some interesting progress suggesting that these features are not defined by functional batteries, not they are emerged randomly, because these features, for example, chandelier cells or layer 5 cortical fugal cells are reliably generated in each individual of the species. There has to be ultimately a genetic program that generate a set of functional genes, such as cell adhesion molecules, ion channels, synaptic proteins. And these are most likely orchestrated by transcription programs, as we have heard from Kui already, transcription factors that we have found before is very important, but now it's also very important in the whole brain transcriptomic data sets. But these are unlikely to be, again, random. They're likely to be a gene regulatory program that ultimately is shaped by epigenomic architecture through developmental programming. And ultimately, this cell relationship and the communication is the ones that underlie the circuit computation. So we would like to think that this communication definition has a potential to really integrate in key features and from molecular genetic to circuit function. So the implication is that neuron types may not be just defined, may be defined by their relationships in the circuit, how they communicate, not just cell intrinsic, cell autonomous properties such as their shape or their intrinsic properties. But more importantly, that these communication features are in fact, or have to be encoded in transcription signatures guided by a gene regulatory program. So the problem is, despite our incredible progress in single cell genomics and morphological analysis, we actually don't have a way to measure connectivity and communication. So connectivity requires EM connectome, which is actually on the horizon, hopefully. And if we borrow the lesson from Drosophila, when the connectome, the higher leucine connectome emerges, I think the cell type at that level will pop out when we recognize the connectivity motifs. But even that may not be enough, because ultimately it is functional connection. It is the transformation of input to output during behavior that ultimately define the unique communication style. And I don't see how we can do that. It'll probably require next generation technology. But there may be a shortcut. The hope is that these are, again, not magic. These cell types and the way they communicate, at least for the cardinal types, for the supertypes, they are reliably generated during development. They are conserved identical among individuals and probably conserved across species. So there is a genetic program. And if we can use ground truth experimental systems to actually discover the relationship between transcription signatures, including transcription factors, and the underlying gene regulatory programs, how they encode and predict these phenotypes, then I think that these are actually written in the transcriptome and epigenome. if we can learn the rules, then we can actually leverage the very high resolution multiomics data to advance cell type definition. So that's my take for today.
Thanks, Josh. So, so we're going to open it up to the panel with, you can raise your hand for, to discuss issues. I guess the first question I'll put to the panel and hope to get some response to is, if we think of these talks and, the previous ones we saw during the day, one of the themes is, moving maybe past pure quote unquote unbiased, transcriptomic clustering. So on the one hand, we have, sets of cells we're calling cell types that come from unbiased clustering of cells. On the other hand, there's a desire to integrate, epigenetic information, biological information, knowledge of function, cross-species information, and then there's developmental variability. And so I would put to the panel, how do people feel about the ad hoc definition of cell type that we currently use of, clusters clustered by gene expression, and then, with metadata attached to that, or almost do we need new language so that when we have new modalities, they can be incorporated and we can call those things cell types. So is the ad hoc definition of cell type satisfactory or not? Are there any thoughts around that? So again, we just heard a series of talks where the pivotal achievement of many of us, contributing or, other groups is that 5,000 cell types in the brain. Are you comfortable with that claim? Or is that a transient claim that is in progress, or, it's based principally on gene expression data? Are you comfortable with the claim that cell type can be defined principally from gene expression at this time in the brain, let's say?
Can I start?
Please.
Jesse, yeah, that's a great question. I'd say, the claim of 5,000 cell types in the brain is really approximate, right? And it's just I think it's a lay language, so that people know what we're talking about. But strictly scientifically, in the papers, we always say very clearly that those are clusters, and particularly they're transcriptomic cell types. And I think, like in a paper, when we say cell types, we define all levels of cell types. The cell types, classes, subclasses, supertypes, and types. They are all definitions of cell types, right? It's just like the granularity is different. That's why we differentiate them at cluster. Yeah. I mean, it is a very good point. I think it's hard to convey that to the general community when you talk about cell types. So I think it's a question. I just wanted to start with a comment.
No, for sure. I'm trying to be a bit provocative to get people talking, but I will say that if we're not totally happy with that as the perfect Platonic definition, what is the right definition? So anyway, I'll Trigva, if you have some comments.
Yeah, at least we say it's transcriptomic cell types.
Right.
We're not saying this is the ultimate cell type. Yeah.
Yeah, I just wanted to throw out there, I think something that's probably in the minds of others is that evolutionary conservation over millions of years, I think, points to the functional significance of these clusters of cell populations. So that's, I think, one piece to think about. It's the technology we can apply at scale now, and so it provides a framework to then move towards maybe a fuller definition. So I think we have to start somewhere. And then the last piece is, even if you don't think these are, every transcriptomic type is a functional unit, there's a genetic or epigenomic hook to then target that type and study its function and potentially treat disease. So.
Right, agreed. I think that harkens back to some of the debates in ENCODE even, where the fact that something is molecularly useful doesn't mean it's functional in the sense of conserved, but it still can be useful. I'm a big fan of the conserved as functional definition, so I'm partial to that. Sten?
Yeah, if I can jump in, I would say that it's important to keep in mind that clustering is a way for us to organize this high-dimensional space, and whether it's 5,000 or 2,000 or 10,000 is always going to be a bit arbitrary. But one of the, I think, important observations from the large-scale atlasing of the brain that's been going on is that, to a very large extent, the clusters at many levels reflect developmental origins. And so I think that should be one of the primary organizing principles as you build the taxonomy of adult cell types, to align them with their developmental origin as much as possible. And that will be possible to, up to a point, up to a certain level. And beyond that, you will have, things that are affected by hormones or local gradients or activity and so on that go beyond developmental origin. I think it's important to at least anchor the framework for a taxonomy of brain cell types in development. And also that gives you a nice transition between actual developmental cell states and the adult cell types. You know, there's a common language between the two if you organize it in that way.
I'm curious, just to follow up on that a little bit, if there was a developmental feature that was like mechanistically it's real, but you don't think it has like functional significance, meaning it's a spandrel, you know, in development, and it produces a cell type that's distinct, you know, molecularly, but there's no conservation. So I'm just bouncing it against Trygva's suggestion. Would you want to call that a distinct cell type or not? Or is that just bad use of language at that point?
If it's not even evolutionarily conserved, then I don't know. I mean, then, yeah, I mean, it's still there. It's an observation. It doesn't mean that, I mean, then you can be then you should be very skeptical that it has a function. It could be a human-specific adaptation, obviously, but I think those are rare. And I think if we get everything else right, we can worry about those towards the end of the project.
That's good. Thank you. Leor?
Thanks, Jesse. I appreciate your question. And I'd just like to focus in on something you mentioned, which is the integration of different modalities and how that should play into the definition. What I think is worth pointing out is that there is no real well or clear notion even of transcriptomic, because transcriptomic already involves multiple modalities. What I mean by that is that, for example, when we perform RNA velocity, we actually generate two count matrices, one from reads that are aligning to introns and the other to junctions or exons. And we really can build two count matrices for nascent or proxy for nascent and mature RNA. And if you cluster the one matrix, the nascent, you get different clusters than if you cluster the mature. Similarly, when you think about single nuclear RNA-seq, and this came up earlier, you're already also looking at really a different modality. It's you know, just a different measurement. So I think what we need to think about is, you know, how to integrate modalities starts with how do we integrate just transcriptomic modalities in a coherent way to even just to cluster them. And I think that has to happen via some kind of biophysical model for how they relate to each other. And so that's my view, is that absolutely integration of all the different modalities is essential ultimately for a molecular view of a cell type or even a cluster. And that starts with thinking about transcriptomic data in terms of its multiple modalities.
I see. So just to follow up on that a little bit. So like typically, I guess people, when they look for cross-modality integration, in a sense, they're just testing robustness often, right? They're just saying what's kind of the dominant signal across a lot of data, but you're proposing something, it sounds like much more sophisticated than that, where the differences between, you know, nuclear versus, you know, nuclear and whole cell RNA-seq would actually be accounted for in the model itself. Is that, and do you think we're in range of achieving that, or is that like more the long-term, you know, thing we should aspire to?
No, that's what I'm proposing, and I think we absolutely have the kinds of measurements we need to achieve that. Yes, I do.
Great. Benna?
Yeah, thanks. What's so interesting about your question to me is that, I mean, yeah, we have an assertion of 5,000 clusters, which might be revised with more data. But we also have, we've heard in this session, and I think it's reflected in several of the BICCN papers, not just Honghui and Zhizhen, but Jonah and Evans, is a specificity of a gene category, and specifically transcription factors. And there was a lot of interest in the Q&A about that early on. And it seems like that could be a strong and testable hypothesis. And it has a long and rich history in specifying the definitions of cell types in other systems. Like if you look at Oliver Hobart's work at Columbia and C. elegans, he would say it's not just transcription factors, it's like specifically homeobox genes, and there have to be 2 or 3 of them in a co-expression set in order to define a cell type. So I'm just curious whether with with the interest in transcription factors as a class, even to define adult cell types, do we say, okay, well, 5,000 clusters, we don't know if those are types, but we have this strong idea about transcription factors. Are we willing to say we can now predict whether cell types are conserved or whether they're real or not, based on a strong assumption that the transcription factors have to be shared, and that they would then give rise to these other functions and other data modalities that we might measure to look at the consequence of that. But I'm just curious whether we are there, at least in that code, which is not just data driven. It does a strong assumption.
Right. I mean, so I'll certainly let other the panelists chime in. And I won't put to you the same question I put to Sten, but the way I think about it a bit is, if that were in conflict with some other ad hoc definition, like, is this simply a useful approximation of the way we think about things, which could be ridiculously useful, so useful no one will ever think about it another way. But if it was in conflict with some other definition, like conservation or development, which would we ultimately say, like, if we just knew from on high, someone told us, nope, super useful, mechanistically it happens, but it's not conserved or it has no developmental origin or something like that, which would we adhere to? So I guess my own thinking, again, I'll let the others chime in, is like TFs are so mechanistically important and central that we'd be foolish to ignore them, but it's probably not my own, if I were, my platonic definition of what a cell type is somehow. Wrong.
Thanks, Jason. Yeah, so this is very good discussion. I enjoy so much. I want to kind of bring your attention to one of the papers I read. I put it in a chat box from BICCN and Barbara Truland. So this is a very intriguing philosophical perspective and what is cell type. And I very much agree with what Stan pointed out. I think evolution is conserved, but on the other hand, also in BICCN's paper that mentioned whether or not that should be defined as cell type, you really need to look at the perturbation, how if you kind of perturb that cell that stay in that particular type or state, that probably can be considered as kind of stable cell type. On the other hand, I really think cells are very dynamic or plastic. In immunology, we call that plastic. And that's not a problem. Actually, I'm a physical chemist by training. We often call that thermodynamic states. I think that's totally fine. I don't think that's a problem. Maybe in our community, should we kind of welcome more this kind of cell state rather than always try to find the cell type, right? So maybe supplement our atlas with a lot more cell states that can actually help better delineate the map or atlas. Yeah.
Right. I guess, would you be happy with those being predefined? So one of the things we'd like to have is a deliverable, even out of this discussion, maybe. And so if there were like an ontology of cell types and cell states, even ad hoc ones that could be attached to various types in combination, like, is that one useful, way of having at least some information to attach to the defined clusters that we see? That, and I'll put that to Ron specifically since you're just chatting about it, but.
Yeah, very quick response. I think, so I think the state means the cells can really change their functional activity or something like that. That can be, I don't think we can perturb, okay, if we are looking at the human brain, okay, maybe organoids, that works. But combining data from different modalities, different layers of the omics, you do get some level of dynamic or kinetic information that can, to some degree, infer the states. I see Fabian is next. I think Fabian should have a lot more insights about that. Okay, I will hand over to you guys.
Sounds good. Okay.
Thanks. If I can go ahead, Ron, I was exactly sort of jumping a bit on that dynamic interpretation. I think this one point, like really naive one, I think that sort of in human cell atlas, we also have been discussing for a long time, so it's not a brain-specific thing we've been trying to make, is that the notion of a cell type obviously is a human-made metaphor, so right, that something that we need as an abstraction to think about this complex system that does not actually care about what we call it, right? So, these cells are these dynamic stochastic things. They're stable for some time. They transition into other ones. And I think it's extremely useful to give these names and sort of to build atlas to be able to integrate and then compare things. But I think it's also obviously dependent on what we choose, right? I mean, we talked about evolution conservation, like which modality to choose, right? I mean, if in the end a cell type is what we call a cluster and we accept that, then it's dependent on the data, the distance, but then maybe also the resolution. Remember, right? If we do clustering, it matters which resolution we give the thing, right? If you keep clustering, sort of keeps getting more fine-grained. So if you take a very high level, like a very coarse-grained resolution, then we get what Honkui called, I think, maybe level 1 annotation. So very high level, right? Maybe immune cells versus neurons, something. But then you zoom in and you keep clustering up. I think it's okay to accept that. I think in the end, if we want to build a map, maybe people call things a bit differently, but you need to know how to map between these. So I think that's maybe in the end crucial. if you want to bring together all these different brain data sets that are being generated in this consortium, maybe it's good to agree on either the same notation or a way how to map between, and then maybe, you know, Stan has these, I don't know, 1500 or whatever cell types, someone has a bit more coarse grain, and then you can maybe map on a certain level.
Right. I think that's an excellent point. I'll give time to our last two hands raised, but just to amplify maybe that, like, I think the key idea that we need to agree on definitions, but they are in some sense, or maybe in every sense, arbitrary. So it's more about agreement than anything else. And so that they're almost not to be argued, you know, except their degree of consistency with previous use. But that, like, if we could just agree on what we all mean when we use certain words, I think that would actually go a long way towards, you know, allowing people to conduct analyses that are meaningful, you know, to everyone. Thing.
Yeah, so I just want a quick comment. Like, if we say typing something, that means that we have some static view about cells. So, but in reality, actually, cells need to interact with each other. So, like, even the same cell may function differently when they interact with different cells. So, I pretty like the George's presentation about cell-to-cell communications to see if we can have additional modality data or dynamic data to really match the multimodality or gene expression to those communications, like cell-to-cell interactions. So that might give us better chance to understand the cell functions or cell types. Yeah.
Absolutely. And the last word to Basilica.
So I'll try to be quick. So first, I'm a big proponent of conventions and unified language, regardless of what that may actually mean in terms of the biophysical world and biophysical definitions. Meaning we need to use common language to be able to talk about same groups of cells. That also means we need to use similar methods to define them and things like that. So that even there could be disagreements, but I feel these operational definitions are essential because we need to be able to talk about things. I think type versus state is one of the things that frequently gets people talking. And I think it's sort of, when we measure transcriptomics, we measure both. And we can decide, again, arbitrarily what we will call one or the other, because maybe we'll use activity-dependent genes to define more types, more states versus types, and et cetera. I think these discussions are important, but I don't think they are initially essential. Initially, a common language is really essential, that we can refer to groups of cells in the same way. I think really to distinguish this type versus state, we will need detailed biophysical models. We'll need to test reversibility probably for states versus types. And I think that's, to me, that's long distance away. I'm more for utility, common nomenclature, common names, and try to get to that. And these philosophical things are useful to discuss, but I don't think we will resolve them that quickly.
I guess the actual last word is going to go to John, since I can't.
Sorry, I didn't mean to do that to everybody. Yeah, I agree. Look, this is a great conversation. I agree 100% with what Silke just said, so I can cut my comments in half. I mean, we do need, there's a practical aspect where we do need to be talking the same language so people can work together in a way that doesn't conflict too much. But I put a comment in the chat. I couldn't access the Q&A. I view these clusters as just hypotheses to be tested. I mean, let's be real. Let's not take our data too seriously until we actually can validate it through some independent means. Now, you could see validation with other omic modalities. That's certainly fair game, or functional. One could also look at this whole question of cell type versus state as a type. What we might agree upon as a type might reflect some kind of self-stabilizing state, which could be robust to perturbation. So, the beauty of all this is if we can get everybody kind of talking a similar language, maybe different dialects, then people can start asking these questions and designing experiments to test. Is this thing that I'm calling cluster, is that actually reflects something that's stable and has a specific function? So I just, as much as I love all this stuff, I just always have to remind myself not to take our data too seriously. I mean, obviously take it seriously, but not to read in too much to it until one actually has validation of what the data are actually predicting. And these clusters really are just predictions until proven otherwise, in my view. Very powerful predictions, I might add.
Right. I think that's a good place to end for this mini session, and certainly, I think that something we probably mostly agree on, which is the power of convention, right? That we just need to use the same language at least before we get into the philosophical significance or ground truth aspect. And so I'm turning it over to GC for the second mini session.
Yeah. So it's a great discussion. So we are gonna next point. We're gonna basically extend a little bit what we discussed in the first part and focus a little bit more about the the caveats and in the intricacies of the data integration and harmonization and the annotations. So we have two speakers again, and the first one is Professor Nanan Sestan from Yale University. So Nanan, could you take over?
Yes, can you hear me?
Yep, we can.
Can you see my slides?
Yep.
Okay, thank you so much. I will, first of all, thank you, Jesse and Guo Chen, for organizing this panel and all colleagues for stimulating presentations. I will make this a little bit more complicated because I would like to talk about two topics that I think need to be addressed better in our atlasing efforts. And so first one will be to remind us that the development of the human brain is very prolonged process and that is characterized by transient features specific to cell types and species. And then in the second part, I'll just basically talk a little about when it comes to what's so special about our human brain and the effort that we and others have done, and then basically that we share all cell types, at least compared to our closest relative chimpanzees. But the point would be that all cell types have changed and that basically even those that are highly evolutionary concerned. So as you probably all know, basically the human brain development is prolonged and many works from many over the many decades have shown both histologically and molecularly that basically some parts of the brain extend all the way to the second half of the third decade. There is a lot of evidence from imaging histology of the prefrontal cortex that also correlates with the emergence of higher order cognitive functions, while the other parts of the brain, such as the cerebellum, seem to be maturing faster, including those that are involved in the motor process, such as the cerebral cortex. So basically, is very important thing is that not all brain regions, and for that reason, cell types mature at the same level, and some of them really take. So when we talk about the development is not just prenatal versus adult, it's not just one period of the development, and we have to be very careful when we generalize. The second point the second part of this is that when you look at the transcriptomic differences, work from our lab and many other colleagues here have shown over the last 2 decades that basically if you look at the differences at the level of gene expression, whether it's a bulk tissue, cell type, that these, when it comes to different regions and development, they are neither constant or static, neither they are increased or decreased with age. And work from our lab has shown, many others have shown that basically what it is that the transcriptomic differences are largely transient. Actually, they are the most prominent during early fetal and mid-fetal development, and then they become a little bit less prominent during birth or perinatally, and then they go up again in infancy and childhood. And again, basically, if you find something that is developmentally regulated in infancy, that does not mean that it's really true or generalized for prenatal development. And the same true is for macaques, actually. So this is not an artifact or uniquely human feature. And also, if you look at the evolutionary difference, there is more and more effort in our effort. Also, these evolution differences are also neither constant, static, nor they increase or decrease with age. They also have very similar cup shape with this late fetal transition. And of course, as many of you have mentioned, basically now with the single cell, we can follow trajectories and lineages of cell types. We can patch what we see during the development, but most of those efforts, including those that we are doing, are either focused on set of regions or really fetal versus adult. And yes, we can learn a lot, and that has already been obvious from papers as well as a presentation. But I want to tell you how really, we show one example that we have published many years ago, actually over a decade ago, that really exemplifies how hard the problem is that actually we need to search. And over a decade ago, a postdoc in the lab who has his own lab, Ken Kwan at the University of Michigan, found that actually a unique set of cells in layer 5 of just during the mid-fetal development from 13 weeks to around 23 weeks of post-conception, okay? They express protein NOS1, nitric oxide synthase 1 in pyramidal cells. This is a protein known to be expressed in interneurons. And to make story short, it only expresses in this region that will become a future broke-up speech area, and only during this 13 to 23 weeks of post-conception. And it's a very small. There are only 30,000 of these little columns. Maybe there are 300,000 of these cells. We could deduct that they are, even within the layer 5, they are unique to subtype. And this regulation is controlled by FMRP, gene mutated in Fragile X syndrome of intellectual disability. And he was able to show that even this is regulated in species-specific manner, just to exemplify how it is. And so in essence, what I would like to say is that when it comes to cell types, regional and evolutionary difference, they are neither constant, they are temporally regulated. And in essence, we don't have enough picture of this complexity when it comes to development as well as the cell types. And then Most of the data is now mid-fetal versus adult. Neither of them are representing well. Even in adult, there are changes, okay, across the decades. And then the second point I just want to make something that has already come across and will be repeated probably is that even share of homologous cell types display human-specific, of course, that for that reason, species-specific changes. And work from many have shown that that many of them may alter transcription, electrophysiology, and neurotransmission. And so basically, to summarize from our work, it was done on prefrontal cortex in 4 species: human, chimpanzee, macaque, and marmoset. So closest relative chimpanzee, older monkey, macaque, and newer marmoset. We have 113 cell types, transcriptomically defined, or hypothesis, as John would say, and I do agree with that, by the way. And only one was unique to humans, that the type of microglia. But all other cell types have very relatively similar proportions and ratios, which I don't think is something that is essential for our uniqueness, at least between human and chimpanzee. But all of them have both gain and loss of gene expression when it comes to human lineage. So even the cell types that are extremely old and shared by many, many other mammals are changed, not just across rodent versus primate, but also in 6 million years of our evolution. And that is the main difference. And I'm trying to understand this and how they affect even diversity of cell types and their properties is a separate topic. And here is, I'll finish with one other example showing how we need multimodal data set, because it's not that. So we observed that actually a subtype of interneurons in chimpanzee do not express TH, which thoroughly hydroxylase a critical enzyme to make a dopamine. And the same homologous cell type exists in chimpanzees. And then also we further found out that basically is that even in the human, where they express both TH and somatostatin, they actually cycle between able to make a TH, which is and also ability to make a dopamine and somatostatin. That is not even, that was discovered actually even decades ago with Xenopus. And it seems that this is a conserved mechanism, but for some reason only works in a human-specific lineage when it comes to this subtype of cortical neurons. This cannot be found on transcriptomic level. Basically, we need to look at the proteomic levels. So thank you for your attention. Look forward to the questions.
All right. Thank you for a great talk, Nana. So next is Xiaoyin Chen from Allen Institute. Xiaoyin, please take away.
Thank you. So I'd like to talk about integrating multimodal data from a spatial perspective. So I think many of us are already quite familiar with spatial transcriptomic data, which basically allows us to look at gene expression and assign them to space, look at how gene expression distribute over space. But these spatial techniques actually allows us to do a lot more than that, because space is incredibly convenient as an anchor to assign multimodal data all in the same cells. For example, my lab does a lot of in situ sequencing to look at gene expression and transcriptomic types of neurons in space, but we can also look at genetic labeling in the same cells, either by directly looking at fluorescence or also by using in situ sequencing. And we can also map projections and connectivity of neurons, again, in the same cells by sequencing RNA barcodes. So all these spatial techniques now gives us the opportunity to obtain these multimodal data at an unprecedented scale. But these data sets also highlight the challenge in understanding these data. So today I'm going to talk about some of the challenges and opportunities that we're thinking about using 2 example projects that we're working on. So in this paper, which we just got accepted in Nature in principle, we used in situ sequencing to interrogate gene expression over one hemisphere of the mouse cortex. So this allows us to look at how gene expression distribute over the whole cortex, and we can use this to define brain parcellations by these molecular features. So for example, in the paper, we define these cell type-based modules, which are groups of cortical areas that are more similar in cell type composition. So ideally, if you want to do this, you want to do this across hundreds to thousands of animals to account for individual variations and look for conservations within the population. And I think with the techniques that we and others are developing, we are now actually approaching that capability. But the other thing you can do when you can interrogate many animals is that you can combine this with the developmental perturbation. So as part of the same paper, we did the same thing across 8 additional animals, and 4 of these have undergone binocular enucleation. So we want to look at how peripheral inputs shape cell type development. And as expected, we saw that in the visual cortex, you can see a lot of cell type changes. For example, this yellow cell type in the control animals is quite dominant, but after enucleation, these animals have a lot of these green cell type. And now if we zoom out to the whole cortex level, you can actually see surprisingly that there's a lot of changes broadly across many particular areas. So each dot here, the circle size indicates how many cell types changed in its abundance in each of these particular areas. And then we can look at broadly like what this pattern means in terms of the visual cortex changes. So basically, we can establish causal relationship with brain-wide structures by combining this brain-wide approach with perturbation. We can go beyond gene expression and also link gene expression to projection. So this is a collaboration with Paulina and Jeremiah at the Allen Institute for Neurodynamics. And here we're interested in understanding this, the organization of projections from the locus coeruleus. So we inject a Symbis barcode library into the locus coeruleus. We sequence these barcodes, which allows us to map these projections at single cell resolution. At the same time, we do in situ sequencing to read out gene expression so we can link projections to gene expression to their spatial organization. Now, this is still very preliminary data, but with some simple clustering, we found two groups of projection patterns. So one group of neurons that project anteriorly to the cortex, and the second group that project posteriorly to the medulla and the brainstem. Now, grouping is sort of arbitrary. So how do we know this grouping is real? Well, one thing we can do is that we can validate the grouping by looking at spatial organization. So you can see here, now if we see where these neuronal somas are, the anterior projection neurons are mostly in the dorsal part of the LC, whereas the posterior projecting neurons are mostly in the ventral part. So you can validate neuronal subpopulations defined using projections, using gene expression by their spatial organization. Now, the ultimate goal here is to link this to gene expression. And here we saw a more complex relationship. So here we found 4 transcriptomic types of neurons in the LC, and you can see that all 4 of them have both types of projection neurons. So this is similar to what we and others have found in the cortex before. Basically, transcriptomic types don't really correspond to projection differences at a very fine-grained level. And so this really highlights this challenge of identifying gene regulatory programs that specify projections. And I think perhaps going back to what Nanette mentioned about the developmental axis, this makes sense. And if we look at what happens in development, we can probably get more insights into this. So as a summary, I've shown you that the spatial data provides all these opportunities for generating multi-model measurement at scale, and this brings a lot of challenges as well. And here are some of the challenges that I talked about, and hopefully we can have some nice discussion from here.
Great. Thank you very much for sharing your fantastic work. This is really brilliant. So the spatial biology is very exciting new development in this field, and that's something that I personally very, very interested in. So as far as I understand that most of the clustering, going back to the cell type definition, is mainly using the gene expression patterns identified from the from the spatial platforms. But as Xiaoyin beautifully showed, I mean, this typically it has a lot more additional information there, right? So connectome, cell morphologies, and things like that. And I was wondering, like, for the people who are traditionally thinking about the cell type for a long time, how would you think about incorporating this additional information to help you better understand the cell types? Josh?
Yeah, Xiaoyin, yeah, I think the last piece of data you showed was very interesting. I was thinking about so-called ground truth biological systems, and my definition of ground truth ultimately is connectivity. But as we know, connectivity data is not available in most cases until we have the EM. But projection is a good approximation. So in your locus coeruleus neuron, the anterior projecting versus posterior connection, there's almost no doubt that they connect to different targets, right? So by definition, they really should be different types. So I guess my question would be, we should use that information to guide the clustering rather than using clustering every gene, each has an equal vote to emphasize, transcription factor. But I would say from our previous study, again, based on this kind of information, cell adhesion molecule clearly will play a role for connectivity, synaptic transmission, signaling. So whether one should really use that information to almost like play with or really shape the algorithm until you actually cluster that begin to make sense. That's my comment or suggestion.
Before I move on, yeah, that's great. Can I ask Xiaoyin to, this is brilliant. So can I ask Xiaoyin to briefly respond before moving on?
Thank you. Yeah, I agree that there's a lot of information and projections, and you can definitely use that as one of the validations for transcriptomics. I think the one challenge, especially in looking at projections of neurons, is that people have had more than 10 years of, or maybe around 10 years of time. I mean, people in the field have about 10 years looking at transcriptomic data. And we have never had access to projection data at the scale that we had until the last few years. And so I think there's a lot of challenge in understanding the diversity in projections, like what should we consider as the same types, what reflects biology, biological types. And there's also the projections are shaped a lot by development and by activity, like we showed in our paper. So I think these are some of the challenges that we need to solve in understanding how we can use the projection data.
My quick response, just very quick. I think the problem is that the projection itself doesn't speak. Ultimately, you need connectivity. So, and the projection also, there's a basic scaffold versus the activity-dependent plasticity. So we need to recognize what is the core feature, but ultimately that require connectivity. That's my opinion.
Great, excellent. Honghui,
yeah,
some feedback.
Yeah, I want to comment. This is actually a very, very important point, and it's often very confusing to people. You know, whether which one is more important: projection, connectivity to define cell types, or gene expression, transcriptomics, or genomics view. Right? Is the genomics view? Is the connectomics view? Will they agree with each other or not? So it is a major question. That's why sometimes I don't want to get into the detailed debate, because you need to step back to see the big picture. There will be always disagreement, and there are reasons for that. So I just wanted to say that in our 2021 paper, a morphology paper by Pen et al., we looked at 1,700 single projection neuron projections, and I think across different cell types, really trying to understand like what's the big picture. So again, I think we need to look at this question at a hierarchical level. Actually at the major cell type level, subclasses or even subtypes or whatever, major cell types, there's a high degree of agreement about what is a cell type. Major cell types, transcriptomically defined, they have very, very specific projection patterns overall, and they are very consistent with each other. However, if you go into a particular type, there's tremendous heterogeneity of projection. We're not even talking about connectivity yet. We're just talking about axon projection, because if the cells have different projection, of course they have different connectivity. If they have the same projection, they may not have the same connectivity. So that's yet another level of complexity, as Josh was saying. But there are tremendous diversity and heterogeneity of single neuron projections, even within the same transcriptomic type. And we summarize those variations under three different categories. One is region specificity. A cell type could be shared between different regions. Let's say somatosensory cortex, visual cortex, and they have completely different projection patterns, even though transcriptomically they're similar so far. And the second is topography. As Josh said, this topography rules things in nervous system. Everything is based on topography. Position, spatial organization, extremely important. So, same transcriptomic type located in different parts, they will have different projection patterns. That's number two. The third one is just randomness, stochastic. It could be activity dependent, experience dependent. It's the same population of cells. They will, for whatever reason, they will form a subset of projection, pattern. Could be, possibly due to stochastic reasons. So all these things are in play, together. How do you sort it out? But this is all at a refined level. At a higher level, they agree, projection and transcriptomics agree with each other very well. But at a refined level, how do you sort it out? So Josh's proposal is a good one. You do supervised clustering to uncover molecules, genes that correspond to a particular projection pattern, for example, topography or regional specificity, right? So that's one thing. Another is that transcriptomic profiles is inherently multidimensional. If you do, and that's why, we debate about how many clusters, because there are different sets of genes corresponding to different cellular properties. And if you use all genes, you can get more and more clusters because you identify groups of genes that correspond to different properties. You can get PC 1, 2, 3, 4, 5. We actually recently have a very interesting paper from our PatchSeq group, Stacy, Nathan, and others. So they showed that morphoelectrical properties, different kinds of morphoelectrical properties could be corresponding to different PCs in the transcriptomic data. Of course, those may not overlap with the PCs that you use to separate cells into clusters. Sometimes they agree, sometimes they don't agree. Even if you use the PC1 and PC2 to separate cells into clusters, there are still PC3 and PC4 there that could be corresponding to a different cellular property. So, and do you want to keep splitting cells, using all 5 or 10 PCs or not? That's a choice you want to make. But I just wanted to say that at a fine-grained level, there is heterogeneity. They may not match, they may match, or you can always find specific genes that match to a particular cellular property. Anyway, it's a
Wonderful. This is a great discussion. That's exactly what we want. So having this kind of data, right? So that's available. This is really making this very, very exciting time for everybody to explore this. Basilica, sorry, I misspelled your name.
Yeah, no, you're pronouncing it correct. I have a question for Nanand. This change between the phenotypes, this potential reversibility between somatostatin and TH phenotypes. Do you know the time scale? Because that's sort of an example of, cell type versus state. What do we call it? And again, I think all of these need to be defined with respect to reversibility and time scale.
Thank you, Bosika. Very good question. I was thinking to mention that. That's why I actually showed that example, because in 5 minutes you don't want to raise too many questions, but that was exactly what I was hoping to do, and thank you. So, so the TH starts to appear postnatal. So basically in infancy, the first neurons we could see was in the newborn brain, and there are only very few. And it takes probably through adolescence. So this is something that appears, in mid or toward late of the development. That's why it was missed in all previous studies, because they were focused on prenatal development. The second thing is this. If you now take, we know that these neurons can make a dopamine in vitro, in cell culture. We don't know yet that in vitro, so that's a separate issue. So is there now a new cell type or not? Because if you use a clustering, they do cluster with the chimpanzee. There are 2 subtypes. Chimpanzees have both subtypes, and that has been validated by Ed and Trigvo and others also. But in my opinion, and I have no horse in this, but to me, it's a new cell type because 99.99% of genes are same. It's clearly homologous, but it now can make a dopamine. That's a fundamental shift in neurons' ability to function and create. So just to me, that would not be picked up. And so going to what Josh mentioned, so I think that even small, minute difference that cannot be picked at the moment can lead into a new cell type. On the other hand, you can have hundreds and hundreds of genes differentially expressed between, let's say, 2 species or 2 cell types, but those changes may not be what drives the functional differences in neurons or glial cells.
Thank you, Nana. So one thing I maybe misunderstood is this is not in the same species. In one species, this is SST, and the other is TH, or did I miss that?
So basically is this. So chimpanzees do not express TH, so they don't have a TH in cortex, and nobody knows why, okay? And this population of TH expressing neurons exists in macaque and humans, okay? So it's basically, it was lost in common ancestors of humans and chimpanzees and reappeared in humans, okay? 6 million. So for some reason, it seems to be advantageous, or at least was purified. We could not find a human that does not have it. But even in humans, it cycles between making a TH and somatostatin protein, and also in humans has DDC. So human cells can make a dopamine, monkey cells cannot. So these 2 genes now make these cells that have clear homology in chimpanzee and macaque and even marmoset different because they can make a dopamine. That's a fundamental difference.
Thank you.
There any more comments?
Yeah,
follow up?
I have one if there's time. Is there time?
Go ahead, John.
Yeah, thanks. This is a great conversation. just to quote Yogi Berra, I'm getting deja vu all over again on two levels. And forgive me on the first example, it's been a while since I taught this, but Nanad, your study reminds me of this change in phenotype in sympathetic neurons from adrenergic to cholinergic, depending on the target. And we actually know the signal, it's LIFT, right? This beautiful work, I think, done by Lynn Landmesser and Story Landis way back in the day. Really beautiful stuff. I'm not sure that back then there would have been argument of is this a cell type or state, but maybe we have more information with which to argue. I think Josh brings up a great point about the connectivity, and Honghui, I agree with your answer, but I think we need to keep in mind, these are great biological questions, but we need to keep in mind that not all development is deterministic or not all cues are instructive. There is permissivity in development. I think probably that's the only way you're going to be able to construct a brain with a limited number of transcription factors and other factors. So, the deja vu is we had a lot of these great discussions in putting together that first MOP paper back in 2021. So, given the amount of work that's being done to integrate information from all these different modalities, I'd like to see if we can stay away from just claiming one modality, one feature defines a cell. I think that's, I think this is going to be more complex than that. I think, as Honghui articulated way better than I can. But just to point out that, not all cues are instructive. There are permissive cues, there are permissive processes. And when I looked at the data from those early studies showing that cells that were defined, quote unquote, defined transcriptomically had multiple connectivity patterns. I mean, that could, as Honghui, that could be stochastic. It could reflect a permissive process, or it could reflect processes that were occurring in development that had later disappeared or we couldn't detect well in the adult state. So I think we just need to keep an open mind about what the biology is in order to really sift through these data and really to find meaning in it.
Fantastic. Giorgio?
Yeah, thank you. This is a really terrific discussion. I just would like to add one comment, which is we don't really know the mechanistic details of how function emerges from the brain. We know very high levels of functional specifications of neurons, such as excitatory, inhibitory, modulatory, maybe projection versus local at some high level, Guillory and Sherman, drivers versus modulators. But not very much beyond that at the level of where function emerges from the brain. So I think that I really like what was said by several people. John actually said, maybe the current classification is just a hypothesis that needs to be tested, and it gives us, especially transcriptomics, gives us a very powerful tool for experimental testing. But I would add an additional layer of testing, which is computational. It's a testing by construction. If we really understand how a circuit works, we should be able to reproduce it in a complete model, in a detailed model, and then the cell types really would be the building blocks of those models. So in many ways, the proof is in the pudding. We'll know that we'll have the right cell types when plugging those details in models, we'll be able to reproduce the same functions, or at least approximate those functions. And remember that the correspondence might not really be one-to-one with a detailed mechanism, because nature must be redundant. And we know through multiple examples, there could be many combinations of genes and many combinations of circuitry, even connectivity, that could produce the same function. Certainly many combinations of the input-output functions of neurons and the synaptic signaling. And I think that Josh actually spelled out what the details are for building those models. I think he meant it experimentally, but I think the very same applies to data-driven computational models. We do need at least the potential connectivity, and maybe some of the detail of the connectivity could be experience dependent. It could work stochastically, as Honghui even mentioned. We do need the input-output function of the neurons, at least the ranges of the input-output function, and we need synaptic signaling. And when you put those kinds of details, when we have them, we're quite far from having all those kinds of details, even from a single circuit. But when you put all of those in a computational model, then you can see what kind of function emerges. It's almost unavoidable. At the beginning, it will not work. And then we'll go back to the drawing board, maybe alter the definitions of cell types in order to get a closer match. So I think it will be an iterative process, but I would like to add at least the possibility of computational modeling as a very essential tool in the tool set for finally understanding the building blocks of how the brain works.
That's great. So we do have mini session on the computational tool coming up, led by Jesse. But before we wrap up this sort of mini session, I want to throw out like a question that to me is a very interesting from the spatial perspective. I think we see more than just the one cell, right? We also see its environment. A lot of that's presumably they work together. I guess what's the kind of the next step? How do we move beyond individual cells and see the kind of the cell niche, so to speak, or neighborhood, however people call it? What's kind of the is there like a unified framework like people like to see, like you know, to see that moving on, or?
I can respond that if I may. I think a cell community, but ultimately it's a cell society, right? That's a cell. Right. So to me, if we have the EM, again, I'm glad that this project is funded. I really think it is very difficult, expensive, but imagine if one have the EM resolution connectivity, a lot of the seemingly variability projection patterns actually may be reduced, because even if the axon has somewhat different branches or different angles, they actually may be connected to the same cell. Ultimately, it's the synaptic partnership that matters. So if we can define that, it's very difficult. I really feel it's a huge progress. So it is very difficult to say now, but my prediction is that when we have the connectome, just like the Drosophila crowd, we'll not debate with a lot of these for no end, because we don't know the connectivity.
Thank you. Jim, and then Ron?
Yeah, so I think through the last discussion and this discussion, I feel like one modality that we kind of ignored is the literature. I think, you know, we're all talking about different modality of data, but I think there are a lot of information that's in the literature about like gene relationships, functions, and all those kind of things, where if you take the gene expression data, project that to the literature, extract all the information related to genes, we probably can get a way more information that might be helpful to better define the cell type, as well as to identify the neuron connections and all those kind of things.
Thank you. Ron?
Yeah, thanks, TC. I think I just want to kind of bring your attention to not just the kind of neuronal cells that are connect on the synapse, but also the non-neuronal cells, right? So the glial, in particular microglia, how they interact with other cell types. So that cell-cell interaction is so important. And I think we have the developing brain initiative within this consortium, but we maybe haven't done that much. That's like in an aging brain. So in that case, how the kind of glial cells age and how they kind of produce the factors to impact other cell types and their characterize signaling. So that's so important. I read on how to figure out, but GC, you are doing on a computational stuff. You are really an expert. But I think in the neuroscience field, so one thing I feel that's a little bit missing or a little bit kind of behind compared to, for example, immunobiology. When you look at the characterize signaling or cell-cell contact, the ligand receptors and quite well can identify in immunology, but we don't have that very well articulated, very well characterized in neuroscience. And in parallel, I feel we also don't have the good antibodies or good antibody panel established, because eventually that interaction is not done by messenger RNAs, right? That's done by proteins. Can we kind of develop the protein panel and the antibodies to really map down the cell-cell interaction in that context?
Great, thank you. Because of time, we have to move on. So, Jesse, heading to you.
Thanks, GC, and thanks to everyone. So our last topic is tools and computational innovation. And our first mini speaker is Bo Wang from here at U of T. Oh. Well, I see Josh for sure. Maybe Josh can go first, and I'll touch base with Bo. by email.
He was just on here. I saw a minute ago. Maybe he jumped off, but I can go. Sure.
Thank you.
All right. Hi, everybody. Thanks, Jesse and GC, for organizing this session and inviting me. I want to talk about several ways in which we can think about innovating on the computational analysis front. These are just some examples of things I've been thinking about, but I'm sure there are lots of other exciting opportunities, and would love to hear your thoughts as well. So I think the common theme here in the three projects I'm going to tell you about is basically new opportunities created by having multiple kinds of data at the same time. So the first example is basically thinking about how we can use single cell co-assays to understand gene regulation. So an example of how you can do this in a way that we've been thinking about is with a tool we developed called Multivelo, which extended the RNA velocity framework to incorporate 10x multiome or related data types where you have gene expression and chromatin accessibility measured in the same cells. And so one of the basic questions we wanted to answer with this type of data is, where are the cases when gene expression and chromatin accessibility are actually out of sync or decoupled? And so we developed this differential equation modeling framework for the transcription cycle of a gene that has phases where chromatin can be closing while transcription is still occurring, or transcription can shut down while the chromatin for a gene is still open. And so this model provides a mathematical way of quantifying and identifying places when gene expression and chromatin accessibility are out of sync. This is especially relevant in a developmental context, but I think it is also going to be super interesting to look at in adult cells as well. Another thing that I think there's a lot of room for innovation in exploring is emerging data types where you have spatial coordinates and molecular measurements, but also imaging or morphology features. And one of the earlier speakers presented some of this type of data. So something we've done in this area is developed an approach that we call MorphNet, which can take the gene expression profile of a cell and predict the cell's morphology. And we trained it on several different types of data, including MERFISH, where the morphological readout was nuclear morphology as quantified by DAPI. And we also trained it on patch-seq data, where the morphology is much more rich, dendrite axon morphology. And in both cases, basically the idea is we can train this deep generative model using paired data, where we have gene expression and morphology on the same cell. And then at test time, we can take a new gene expression profile, pass it through the encoder into the gene expression latent space, and then use a generative adversarial network to generate an example of a morphology of a cell that has that gene expression. And one thing that's challenging about this type of relationship is that it's not one-to-one in either direction. So, in an adult neuron, the gene expression profile at any given point in time doesn't completely specify the morphology and vice versa. So rather than thinking of this as an exact prediction problem, we're thinking of it more like conditional distribution sampling, where the more you know about the molecular profile of a cell, the more you can narrow down its morphology up to the limits of how that modality specifies the morphology causally. So I think there's a lot of room for innovation in relating these types of data as well. A last example of how you can innovate in this space in terms of new data types that you can put together is in terms of using the actual anatomical locations of cell soma to infer something about their anatomical properties. And this is a project that we were funded by as part of the BICCN that we're now wrapping up and just about to publish. And what we did in this project was we used our Liger algorithm to integrate dissociated single cell profiles. Then we used spatial transcriptomics data to map the spatial distributions of each cell type. And other groups have done parallel initiatives in similar direction. But the key idea that we were pursuing here is once we know the spatial distribution of a molecular cell type across the brain, we can use that position within a common coordinate framework to learn something about other anatomical data sets that are registered in the same coordinate system. So for example, we took a data set of brain-wide cellular resolution vascular imaging data that was measured across multiple mouse brains, and we co-registered that in the same coordinate system as our spatially mapped cell types. And then we calculated a vascular density score for each cell type and used that to identify cell types that have either high or low vascular density locally. And we were able to create a sort of like a vascular density Manhattan plot where we can identify cell types that rise to the top in terms of their local vascular density. And if you make a spatial plot of where the cell types are located with regard to the major vessels in the brain, you can see that we're getting something pretty sensible here where, for example, this cell type in the hypothalamus is right near the conjunction of several arteries. And one thing that you have to be careful about here, which we thought about extensively, is that you have to rely on the essentially the accuracy of registration as well as the conservation of whatever anatomical data set you're looking at across individuals. And so in this case, we were careful to show that basically what we're seeing is recapitulated across 3 individual mice whose whole brain vascular maps are available. And so I think there's a huge opportunity for this type of integration where you use the actual spatial positions of cells as a bridge to many other data types.
Thanks, Josh. I still don't see Bo here, so I think we'll just move on to the discussion. Bo, you're not here, are you? Not missing anything? Okay. So moving on to the discussion, I guess one of the key questions, there's a lot of methodological elaboration. I could have, many of us could have guessed what Bo might have talked about, which is an interesting and potentially controversial topic, which is, one might think about sophistication of models. But I guess even before that, there's a question of like validation of models and validation of new tools. I guess that's been really key as methods become more sophisticated. If you think of the the wonders of CASP finally being a solved problem, some people might say it really depends on the idea that people had ways of measuring that it was a solved problem. What is the general feeling right now on validation of new methods? Is there a consensus in the field? I'll make it more casual. If you read a paper and you see a method, like, do you know that this is a method you desperately want to try, or it's hard to tell because, it's just everyone thinks their method looks good and it's hard to tell the difference between different methods. So are there any thoughts around validation and comparison and rigor? Like Fabian?
Thanks for raising that. There has been initially not a problem because initially there was all this beautiful single-cell RNA-seq data around 10 years ago, and we didn't have stuff to go around. But then became clear that early tools such as pseudotemporal ordering up to now, or the mapping ideas or multimodal integration became so much fun, so that a lot of, I guess, computational biology, but also machine learning people have been getting interested in these problems. And I think we just counted for single-cell RNA-seq papers that have been around, I think like a year or 2 years ago, it was more than 1,000 different algorithms for particular questions, right? So this is getting a bit of a mess. And I think for even doing up-to-date nowadays analysis, some type of initial benchmarking effort, or if it's sort of an established topic, having something to cite to really give an argument why you choose that particular approach is a bit necessary. Sometimes there's a thing that fits for all. Sometimes you need to be sort of data set specific. But I think comparing and evaluating your tools is something that the machine learning community has been doing for ages, in particular if it's really about small increases in some AUC for classification or so. I think we should be potentially getting more used to that. In a sort of bigger consortium, we've been setting up this open problems website where it's like these Kaggle competitions where you can upload a few of your questions and then have an open discussion about how to even just measure how good potential things are, because of course, depending on the metric that you choose, some things perform well, some don't. And yeah, that's part of the development, I guess. And then of course, in addition, also the typical data sets. And we've been running a bunch of competitions at recent Europe's conferences for various things, from data integration to temporal trajectories to perturbations. And I think there's a lot of interest in those data sets, and those sort of questions you establish there, I think can be really a great asset for the community. So I think some of these spatial data sets being generated here, such as this beautiful Murphy's one from Hong Kong, could become also for the more machine learning people an asset for people to actually evaluate then tools and ask rather standardized questions that non-experts in that particular biology could also then use up downstream with methods. So I think that's also maybe a trajectory that the consortium could take.
I absolutely agree. So I actually think that's a really key point, that the existence of this reference data is potentially solves this problem, right? In part, at least, that this is the data that can be used to evaluate methods. And it's just a question of coming up with, consensus standards and consensus metrics as takes place in any field. Christina.
Yeah, thank you. Yeah, I totally agree. And I think the efforts to try to make a standardized framework that we think about different types of problems is really important. And also taking, the strength of the community and proposing, potential evaluations that will help us to have a benchmarking framework that makes sense. I think one thing that I really thought would be good to emphasize also is just to make sure that the evaluations we're doing and that we decide on as a community are really based on biologically meaningful tasks, and trying to understand, what higher order information or predictions we can make with these models that are not, that are going beyond kind of like some basic pre-processing, but also, new biological insights and, what we really are able to achieve with the huge amounts of data that are now becoming available to train some of these more advanced models. And I think, just one thing we should be cautious about, just to make sure that we don't get really, really good at training models that perform a task that's not, what we're actually interested in biologically. So making sure that we keep that in mind. And then the other thing to keep in mind, I think, when establishing these benchmarking frameworks is to ensure that the problems are sufficiently challenging. So, there's a lot of problems that, you might not necessarily need some very, complex or advanced approaches to accomplish. But, in some cases, then everything look like it's doing the same. But if you actually increase difficulty of the problem, or you get to some of these higher characteristics of genes, or cell states that we would like to explore further as a community, then it will become more clear, if there's certain models that perform well in certain areas versus others. And also we can learn from that as well, so that we can, take the best of all these approaches and put them together to continue to evolve the methods that we're developing.
Right. I just highlight like papers that are now getting old, but when people benchmarked, Sonesson and Robinson, the differential expression methods, I mean, it's pretty striking. The top couple of methods are, like the Mann-Whitney test, stuff like that, or the t-test was like second or something like that. So, that highlights kind of both aspects of what you're saying. Like, is that just because the problems just aren't hard enough at that point? Or the differential expression is such an relatively easy thing that the 20 or 30 more sophisticated methods aren't essential, or it could be that they're overfitting to, when they validated it themselves, they were overfitting to specific data and it's not robust. So I think those are key points, I think.
Yeah, so I'm totally second to that. We really need validation data to validate our computational or machine learning method. Because to be honest, right now, many of those data we got to train our machine learning is like a snapshot data. So of course, we can always try to learn some latent representations or embeddings, but that may be just driven by some like a particular snapshot. So that's why we really need to get some like sort of dynamic data. It's already like a computer vision versus like individual images, right? So if possible, if we can get some high resolution dynamic data, we can use to validate our machine learning models. So in addition to the those dynamic data, also like maybe we can get some additional mechanistic data, like a cell-to-cell communications or the interactions. So we can try to maybe explain some like latent representations what we learned from the snapshot. So try to give people more mechanistic insight at least. Yeah.
It feels like just defining the problems really is something that is key at this point, but then also establishing a framework for validation. And again, not to harp on CASP is maybe something that's just been, I feel, extremely successful. And the key feature of CASP is that the data actually is true validation data, right? It's not cross-validation. It's not, test our reproducibility of our method on this data and it looks like it's robust. It really is held back data that experimentalists generate, and then you have to predict. And I guess, can we aspire to that at all? We have, even, there are ways of doing it like in CAFA, which is, inspired by CASP. Yeah, the data isn't generated for, it's a functional annotation exercise, isn't generated for the teams. It's data that they know will be generated in the next year regardless. And so I think given the timeline of data generation in, among this consortium in BICAN, is it possible that we could set out tasks that would serve as validations for methods based on incoming data that we know is going to be, multi-human data or something like that? Is that something that would be interesting to people, or is that possible? No enthusiasm for that. I mean, we have spatial data presumably that's planned to be generated. It just seems like that might be a mechanism for people to validate, or integration data. For example, if we know there's going to be multiome generated later, well, I guess there's two sides to it. Would methods people be enthusiastic about having their methods validated that way, and would experimenters
How would that be
willing to-
validation? Maybe I'm not getting the point here, Jesse. So you're saying you predict like another modality on like this unknown region, something like that, and then someone else measures it?
If you had something like multiome, for example, where you did an integration and you have some claim pre-multiome, you know, like I'm harkening back to something that now has occurred.
Yeah.
But let's say in the pre-multiome data, we were integrating ATAC-seq and RNA-seq, and we were making claims about that. And nowadays, you just do a multiome experiment, and you might consider that a validation of the integrations that people were doing. We saw some work, you know, people were discussing imputation. Imputation is, again, I think the tasks would have to be defined that create space for validation. But as an example, imputation, which is kind of key, particularly for development, where presumably you're not going to sample, you know, can't sample everything. And then someone is going to sample this area or this time point you haven't previously sampled, and you've inferred some sort of change. So I don't have a clear idea of exactly. I think it would have to be there's have to be some marriage between the tasks that people feel are natural ones and the data that's being generated. And if that doesn't exist, maybe this isn't a good suggestion.
So I agree with some of your points. So let's say if it's a time series and sort of drop out a few things, or you go sort of in future time points, that's a very clear question that a generative model should be able to extrapolate to predict or maybe to go there. One key problem, of course, of many of our generative models at the moment is that, you know, there's no unique true answer. There's always variation stemming from all kinds of sources, which we haven't fully delineated. That could be in many cases technical, but many cases also sort of sampling location dependent, changes across. You know, we're not working in a cell line, right? So individual variation, I don't think we have fully mapped out. So even though you have then a prediction for a particular time point, are you within that sort of error boundary that you would want to have or not? I don't think we can fully answer that. I think it's a similar problem for many of the perturbation predictions that are happening, where, you know, you CRISPR out a particular thing, and then you see some effect, but it's a complex one, and there's technical variation superimposing that. So you approximate it, but the scores that we have for that often, you know, R squareds or mean gene expression differences, something like that, and maybe not the most fine-grained measure for that yet.
Thanks. JC?
Yeah, so I agree. Some of the tasks, they are more harder than others, and some of them are less well-defined, so it makes it even harder. So I guess some of things that are conceptually would be really easy to check, at least, is that A, deconvolution, for example, and B is like, for example, the cell type mapping, right? So when you have, these are the things that are commonly used in this community. And, you know, to my knowledge, there's not really good sense like which method, I mean, how well they really work in the real setting.
Yeah, I think deconvolution particularly, you know, if you've been around long enough, you've seen 1,000 deconvolution methods and maybe a quarter of one validation on any of them, kind of. So like deconvolution feels very popular when you talk to, I mean, when I talk to wet lab biologists, I'm not one. It feels like they actually want deconvolution a lot. Single cell data seems like something that offers a lot of opportunities for new methods. There's more validation in papers now than there used to be, but it feels like, you know, people's methods work on the data they generated. There might be some degree of overfitting. Maybe that's a space for, you know, actually solving a major problem, which is what methods are robust and generalize and things like that. So I guess a final topic, since we have 1 minute or two left, just in honor of Bo Wang, who didn't make his talk, but like how people feel about, you know, more sophisticated methods, like, you know, like foundation models and things like this for, like, is this the aspiration for methods development at this time? Or, you know, are these methods in some sense, like any method which you then need to have an explainable, you know, there's a whole other aspect of the research that's then on explaining the method itself and how it works. It may limit its utility for problems where we really do want concrete, you know, features that are associated with different aspects of neurobiology. So I'd be interested in people's thoughts on enthusiasm for, you know, highly sophisticated, let's say, AI methods at the time. Fabian, I assumed had something to say on this too. So, yeah.
It's okay if I can briefly mention that. So sorry for jumping in. So I think, as in many fields, it's going to be hard to beat deep learning models for complex integrations. You know, this is just the flexibility and the power with the large scale data that we get. You just won't beat it. We've been running, I think, 3 times now NeurIPS competitions, and most of the winning integration methods, if you really go just in terms of scores, were nonlinear, typically deep learning based ones. So our more traditional KNN graph based ones just hard to beat. Having said that, it's going to be hard to interpret, but if you really care about accuracy of prediction, I think that's the way to go, and your interpretation methods are catching up. But that's, of course, one side of the message I'm really looking forward to seeing what maybe, Josh, you think about with sort of also NMF-based ones often are performing very well. It's hard to beat a linear model in many cases, right?
Yeah, no, I think I agree. Ultimately, the deep learning approaches are going to win. I think there's some real challenges in biological data as opposed to text and language, though, especially in terms of the noise and our ability to understand intuitively when the models are getting it right. So those would be my comments. I think ultimately it will be very exciting, but there's still a lot of challenges before we get there.
Great, and I think if you could just, and then Christina, very quickly, since we just
before we go to those complicated models like deep learning or language models, like I think at least we can use some prior knowledge, like biological knowledge. We know, for example, some TA maybe regulate some gene. We use those knowledge to regularize our current machine learning models, might be useful as well. Just a quick comment.
Thanks. And Christina?
Yeah, totally agree with everything I said. I just want to mention also that ablation studies are a way that you can also improve interpretability. And then also at the end of the day, I mean, I think we're ways away from going straight from one of these models to a clinical trial, right? There's a step in between where we would be testing some of these predictions. And at the end of the day, if you think about the astronomical number of possible things that you could test in the lab, this is a way to really hone down and prioritize what are the most high yield possible experiments to do that's more cost effective than testing everything in the wet lab, and then hopefully get us to a more accelerated, therapeutic, et cetera, output from these modeling approaches that are integrating large amounts of data.
Thank you. And with that, this session ends. We won't sum up now, but we'll sum up tomorrow morning. And I'll just thank everyone for participating and everyone else for listening.
Yeah, thank you very much.
Thank you. Thank you so much for such a